Nucleotide excision repair in eukaryotes. This process is again a molecular mechanism that is conserved across several species, including prokaryotes to eukaryotes. However, the molecules associated with the mechanism are different in eukaryotes when you compare it with the prokaryotic system, but the mechanistics is more or less the same. So let us look at the learning outcomes of this session. So, NER is definitely an enzymatic pathway that recognizes and corrects a wide spectrum of structural anomalies, which means we are talking about DNA lesions, and in this case, definitely bulky adducts that are able to distort the helix as well. So, when you look at base excision repair, it is repairing those DNA lesions that do not really affect the duplex per se or the DNA helix per se, but in this case, the DNA helix is, of course, affected, and there is a distortion or there are distortions in the DNA helix, and it is these distortions that the NER is able to recognize as a damage point. Now, NER has subforms. That means it can be what is called as the global genomic NER and transcription coupled NER, with the latter, that means a transcription coupled NER, carrying out repair in transcriptionally active genes. Now, NER involves the excision of oligonucleotides, within which is present the damaged nucleotide or the bulky adduct, and it is found to be majorly involved in repairing damages due to UV exposure. NER follows a sequential molecular mechanistic to address the damage, and therefore any defect in NER has been associated with several different diseases. So, a glimpse of NER in eukaryotes. So we know that, or there are many reports that have suggested that defects in NER has been associated with a disease which is called as xeroderma pigmentosum, a genetic disorder, wherein the NER constituents or components are either mutated or they are less functional. And what has been observed is that those patients suffering from xeroderma pigmentosum are at a very high risk of skin cancer due to exposure to sunlight. The reason being that UV induced damages are not being repaired as the NER mechanism is defective. So this is basically something that was the reason behind, or studying xeroderma pigmentosum allowed recognizing many of the molecules that are involved in the NER pathway. And so most molecules or many crucial molecules in the NER core reaction mechanism are labeled as XP, XP standing for xeroderma pigmentosum. Now, we all know that NER can basically exist in two subforms, okay? One that is called as the global genome NER. This pertains to the fact that the nucleotide excision repair can take place in any part of the genome, whether there is transcriptionally active genes or genes that are not active, not transcribed. So this is something that is basically covering each and every part of the genome. While there is another subform which is the transcription coupled NER. In this case, the basic repair is present in the regions of the genome where there are very highly transcriptionally active genes. So it is when transcription is going on that the transcription machinery comes across lesions, and those lesions are then being repaired by the NER system. Now, of course, one of the first steps in the core NER reaction is DNA damage recognition. And to look at the fact that there are two subforms, that is the global genome NER and the transcription coupled NER. Uh, for both the systems, because there are two different subforms, the molecules that actually recognize the DNA damage are different. So in global genome NER, one has the XPC RAD23B complex, which is responsible for recognizing the damage, while in the transcription coupled NER, you have two other proteins called the CSA and the CSB, along with XAB2, that are specific for the recognition of the damage in the TC, that is transcription coupled NER. Once the DNA damage has been recognized by these respective complexes, then thereafter the repair of the NER remains the same. So the core NER system after the DNA damage recognition point in these two subforms involve the same set of molecules to actually carry out the repair. So let us begin to look at the core NER reaction in the global genome nucleotide excision repair. So which are the core factors that are involved in this core NER reaction? So you have, of course, the XPC RAD23B complex, as mentioned just as mentioned earlier. This is the complex in the global genome NER that is responsible for recognition of the damage. What has been understood is that XPC as a molecule has a very high affinity to bind to the DNA, and that allows the interaction of the NER with the DNA. So the binding of the first molecule that, or the first complex that binds to the NER, is because of the XPC RAD23B, and XPC itself has a high binding affinity to the DNA, which is positive for the NER, because only when you have the complex binding to the DNA, then it is able to identify the damage. Now, the next complex of the next core factor of the NER reaction is transcription factor 2H, which is involved in the NER mechanism, but it is also part of the transcription process in eukaryotes. And what is interesting to note is this, that the transcription factor 2H is almost a complex that is made up of 9 to 10 subunits. And of the 9 to 10 subunits, two major subunits present in the TF2H help in the NER process to a great extent. One also has the XPA. This XPA basically is, in fact, the central core molecule that binds to almost all the other molecules in the NER. You have the replication protein A. This is what is equivalent to the single strand DNA binding protein in prokaryotes. And because it binds to the single strands that are formed during the NER repair mechanism, it also plays a very important role in positioning other molecules in the NER core reaction. Then one has XPG. This molecule is basically endonuclease, which is able to carry out incision. You also have another endonuclease, that is the XPF ERCC1 complex. And so together XPF ERCC1 and XPG are able to cause incisions in the DNA. Now, in many of the eukaryotic systems, there is presence of what is called as DNA damage binding proteins. And this DNA damage binding proteins are able to recognize very specific adducts or DNA lesions. One has observed that there are DDBs, UV DDBs, which are responsible for actually recognizing the CPD lesions, that is the cyclic purine dimers that are formed due to UV exposure. So the thymidine dimers or the pyrimidine dimers are recognized by these DDBs especially, and then these DDBs finally allow assembly of the entire NER system. Now, out of these molecules, the XPC RAD23B complex, the TF2H, the XPA, and the RPA form what is called as a pre-incision complex. Centrin 2 is a molecule that is actually stimulating the XPC RAD23B complex to bind to the DNA. So it is able to get activated through a certain signal transduction pathway, and as it gets activated through a signal transduction pathway, it is finally able to stimulate the XPC RAD23B to bind to the DNA. XPA is a zinc metalloprotein which has an exclusive role only in the nucleotide excision repair. You would not find XPA working in any other repair systems. It is only working for the NER mechanism. And this XPA is able to recruit many other molecules of the NER in the process, enabling the repair mechanism to happen. Now, TF2H, as mentioned before, has two subunits, XPB and XPD, and these two subunits have helicase activity, and they also have ATP binding domain, which binds to the ATP and also has the ability to hydrolyze the ATP because they have inherent ATPase activity. Now together XPB and XPD are able to unwind the damaged DNA duplex. So where you have the damaged DNA, that portion of the DNA gets unwound. So the DNA opens up, and then on both sides of the lesion in the open DNA duplex, incisions are made by the XPG and the XPF ERCC1. Now, going further and looking at more detailed understanding of how the GGNER works, you know that you have whenever you have the lesion present in the DNA, it is first the XPC RAD23B complex that comes and binds to the um, DNA lesion. Now, as you can see that you have two parts of the XPC RAD23B that are able to interact with the DNA. One part is binding to the undamaged DNA, and the other part of the XPC is able to bind to the damaged DNA. So let us understand how this structure enables to do so. So before that, just understanding that XPC RAD23B is the main damage recognition complex. It recognizes diverse damages. This is very important, and that is why the NER is a wide spectrum DNA damage repair mechanism. It is able to recognize any kind of DNA adducts. It can recognize thymidine dimers. It can recognize 8-oxo-G. It can recognize alkylated bases. So you can understand that it is able to recognize so many different DNA damages. And that is the beauty of the NER system, that it is specific, and yet it has it has a broad spectrum of actually being able to repair these different damages. And what is important to note is that these regions where there are damages are recognized by the XPC RAD23B because at these damaged sites, the duplex is unstable. It is thermodynamically unstable. So how is the XPC23B recognizing the damaged region? It is not recognizing the lesion really, but it is only recognizing the fact that where there is a lesion, the DNA duplex is destabilized. And that is what is getting recognized by the XPC23B. To get a closer look at how XPC23B works, you can look at the fact that the DNA that is present has a CPD damage. That means we are talking about a pyrimidine dimer. And interesting to know that the green strand that is present is the damaged strand, because that is what contains the DNA lesion or the bulky adduct, while the other strand, which is the gray strand, okay, is the one that does not have any DNA lesion. And interesting to note, therefore, is this, that the XPC RAD23B complex is not binding to the damaged strand. It is only binding to the non-damaged strand, and a part of it is binding to the undamaged DNA per se. So, the blue domains is formed by what is called as trans glutaminase homolog domain and beta hairpin domain 1. Now these two domains together are able to bind to the DNA, but this DNA that is there is an undamaged DNA. So this part of the XPC will bind to any part of the DNA, any part of the double-stranded DNA where there is no damage. So you can see that the XPC has an affinity to bind to the double-stranded DNA thanks to these two domains. But the attachment to the damaged DNA is not because of this domain, it is because of the other domain present over here. Now, RAD23B or RAD23A are just present bound to the XPC to stabilize it. So, as a stable complex, it is able to bind to the DNA properly. There is no direct interaction that the RAD23BA has with the DNA. It is only the XPC component that binds to the DNA. These domains are basically made up of beta hairpin domain 2 and beta hairpin domain 3. And these two domains are able to bind to the nucleotides that are present opposite to the adduct. That is the here the adduct, of course, is the thymidine dimers. Now, effectively, the understanding therefore is that this domain is not binding to a double-stranded DNA, it is binding to a single-stranded DNA, because at this lesion point you have, because of the thymidine dimer not binding to the nucleotides present opposite, you will have the DNA duplex a little distorted or a little open, and hence this binding um, of the nucleotides that are present in this region where there is a lesion, the domain is able to rotate it to bring it to bind to the BHD2 and BHD3 more closely. This accentuates, or this brings in a kink in the DNA at this point, accentuating the fact that here is where the damage is. So, interesting to note therefore is that because it binds the non-damaged DNA, which is almost single-stranded DNA due to the lesion not being able to bind complementarily, okay, you can look at the fact that because it binds to a distorted DNA, okay, it recognizes unstable DNA duplex regions. It is not directly binding to the lesion, but it binds to the opposite strand which does not have a lesion, but the opposite strand is distorted because the damaged part of the DNA is not able to hydrogen bond with the nucleotides present in the opposite, and that is what is recognized by the BHD2 and the BHD3 component. So this is how the damage is recognized by the XPC23B complex. So we talked about the fact that many adducts are recognized by DNA damage binding protein. You have B1 and B2 being present, and as you can see, you will find that the DNA, okay, is bound to by the DNA domain binding 1 protein and 2. It has very high affinity for UV DNA damage. It is definitely on binding to the UV DNA damage able to then recruit XPC. So in case of, primarily one understands that NER has a very important role in taking care of all damages that happen because of the UV exposure. It means that there has to be something that is specifically recognizing these regions where there is a UV exposure based DNA damage, and then there is something that is calling in the NER system to repair the adduct. That is the the pyrimidine dimer that is formed. So effectively, although XPC can bind to the DNA very strongly and also identify damages, but in case of the fact that you may have many DNA adducts formed because of UV exposure, so to help the NER system to identify so many damages at the same point, you have the DNA damage binding proteins helping the NER. And once they bind to the uh, region where the lesion is, the XPC recruitment is carried out. So what has been observed is that the DDB2 has a hollow hydrophobic pocket that can accommodate the pyrimidine dimers and also the 6,4 pyrimidine pentacycle that is formed. So these both adducts are rotated into the DDB2 pocket from the helix. And this UVDDB2 is also understood to be a part of a larger complex that is ubiquitinated along with histones. So one other thing, therefore, that has been understood is that DDB2 does play a role in chromatin remodeling, making the region which has the UV DNA damage more accessible to the NER molecules. So that is an extremely important find about the DDB2 protein. Now we look at what happens next. So once you have the XPC23 RAD23V identify the damage, okay, the XPC RAD23V complex is able to recruit the next component of the NER, which is the transcription factor 2H. And this binding of the transcription factor 2H to the lesion, you can see that you have a single strand component only about this much formed because of the DNA lesion not forming complementary binding, and also you can see that a kink is found because of the XPC complex. At that point then, when the TF2H binds, you have the DNA opening up, and also the TF2H can carry out the lesion verification. So let us look at how it can carry out the lesion verification. That there is a DNA damage is being verified, and that verification is done not by the XPC but it is done by the TF2H. So XPB and this is the XPD. These are examples of XPB and XPD in the archaeal system. As you can see, you have within this domain present a motif, okay, and this motif is able to insert itself into the double-stranded DNA. Very important for the XPB to prise open the DNA at this point. So this motif that is present in the XPB is actually inserted into the duplex DNA to open up the DNA. Now, what is important to notice that it is this domain that is able to actually recognize the damage, and because it recognizes the damage, it is able to make a conformational change such that this motif is able to pry open the double-stranded DNA. Now, this part of the domain of the protein holds on to the DNA. So this only has a role of holding on to the DNA, and this domain, apart from having this motif that can insert itself into the duplex, also has helicase activities, and it also has ATP binding domains. So the same is observed with XPD. You have a domain that has the helicase and the ATP binding regions. You have a domain that can bind to the duplex. But what is interesting to note is that it has a domain that can bind to single strand DNA on the DNA opening. So when it opens, or when the XPD opens up, or rather the two together opens up the DNA, it forms a small tunnel through which the single strand that is formed is able to go in. Okay, so the XPD has a tunnel formed through which the single strand can move, or through which the single strand can uh, get into. Now, if the tunnel, so the tunnel is too narrow to allow an adduct, okay, which means that if there is an adduct present, if there are normal nucleotides present, then it easily goes through the tunnel. But if there is a bulky adduct present, say for example a thymidine dimer present, then it being bulky, it cannot enter the tunnel, so it is too narrow to get into the tunnel. And that is how the XPD or the TF2H is able to verify that there is a lesion, because on not being able to allow the adduct to go through the tunnel, the translocation of the TF2H is stopped, so the helicase activity also stalls and the damage is verified. So, therefore, on opening the DNA, okay, and translocating just for a while before finally reaching the damage, it verifies the presence of the damage and also stops the helicase activity. So, there is no further opening up of the DNA that is happening. What happens next? So, once you have the TF2H and the XPC-RAD23B opening up the DNA at the point where there is a lesion, okay, comes in another protein that is called as XPA, uh, RPA and XPG to form the pre-incision complex. Interesting to note is that it is a TF2H which has a small subunit called P8 that is able to recruit the XPA. So XPA is recruited by the P8 subunit of TF2H. Similarly, XPG and RPA are independently recruited by TF2H to this. The moment you have the formation of TF2H, XPA, RPA, XPG complex that forms the pre-incision complex, you have the XPC RAD23B being released from the DNA. This is something that is very important. Now what is present therefore is a assembly that is going to begin the process of correcting the damage. So far what was done is that the damage was recognized, okay, and the damage was verified, and the verification needed opening of the DNA. So that was done so far. Now what is going to be done is actually address and see how the damage can be removed. XPA definitely is the central NER factor because it binds to all the other components of the NER system. RPA is bound specifically to the single-stranded DNA, okay, because it's a single-strand binding domain, but it just does not have the role of binding to the single strand. It is also able to position the XPG properly within the pre-incision complex. Now, the XPG per se, and another thing is that the RPA binds to the non-damaged DNA, and it is the XPA and XPD that is binding to the damaged strand. Now, interesting to note is this, that the XPG on binding to the pre-incision complex does not have a catalytic activity, it only has a structural implication on the entire complex. To get this active, because we all know that XPG is an endonuclease, to get this active, this has to undergo a conformational change such that it is able to bind to the single strand properly to make a cleave, to make a cut. As of now, in the pre-incision complex, XPG, although it is an endonuclease, is not active. Now, what happens thereafter is that once you have the pre-incision complex formed, the TF2H now recruits, the TPA now recruits the second endonuclease, which is the XPF ERCC1 complex. Now, on binding of the XPF ERCC1 complex to the XPA, it is positioned to the 5' side of the lesion. So what happens is it makes a cut, and on making the cut, it leaves the 3' OH free over here. This is strategic. Why is this strategic? Because once it generates the 3' OH group, at that point you would have the pCNA being loaded by the RFC, and if pCNA is loaded, you would have the DNA polymerases coming and attaching to the pCNA to begin synthesis using the 3' OH as a nucleophile. Interesting to note over here is that you could have either Paul delta, Paul epsilon, or even Paul kappa coming and helping in the NER process. In fact, DNA Paul kappa has been associated with NER by looking at the fact that if there are damages or if there is a defective DNA Paul kappa, then it does affect the NER process. So therefore, these DNA polymerases using the 3' OH are able to start synthesizing. And as it starts synthesizing, the strand that was already bound is displaced, okay? And once you have the RPA also getting, once you have the new synthesis happening, you have the RPA having a change in conformation, which will also bring about a change in conformation in the XPG. And now the XPG will act as an endonuclease and form a 3' incision. And that will leave a 5' phosphate over here, which is again strategic because once the DNA polymerase synthesizes the entire thing, then the DNA ligase only has to seal it by attaching the 3' OH to the 5' phosphate by a phosphodiester linkage. So therefore that sealing of DNA is immediately after the oligonucleotide containing the DNA lesion has been removed. So you have the entire new DNA being synthesized and then ligated at this point. So you can either have DNA ligase 1 or DNA ligase 3 associated with XRCC1 that can seal the DNA. Of course, DNA ligase 1 is associated more with quiescent cells, whereas DNA ligase 3 is associated with cells that are replicating. So it has been found that these DNA ligases have different phases in which they are active. So during cell cycle at the replication stage, you have the DNA ligase 3 getting expressed, while uh, when you don't have the replication going on and the cell is in the G0 stage, that time the DNA ligase 1 is involved in the NER process. But what is interesting to notice that once you have the TF2H opening up the DNA, the base lesion is present within an oligonucleotide that is cut at one end and cut at the other end. From this end, it is cut by the XPF complex, and from this end, it is cut by the XPG. So the damaged strand is excised out, and that excised out strand has the has the damaged lesion. And this excised DNA strand, which is leaving the complex, is replaced by a new strand thanks to the complex of the pCNA, DNA polymerase, DNA ligase system. So you have the gap filling by the polymerases and the sealing of the nick by the DNA ligase. Now we go to the second form of the NER, which is the transcription coupled NER. So as mentioned before, the transcription coupled NER requires CSB. The name CSB and CSA, they are two different complexes that are required in the transcription coupled NER process, comes from the word Cocktail syndrome. That is because of mutations in these two proteins, you do not have TCNER functioning, which can result into a syndrome that is called as Cocktail syndrome. So as the RNA polymerase is transcribing the RNA, and when it reaches the lesion, you will find that the RNA pol stalls. To the stalled RNA polymerase comes and binds the CSP, and it is this CSB that recruits the NER. So therefore, the CSB has a role to play in identifying the damage. Okay, when it identifies the damage, it's binding to the RNA pol is enhanced, and post that, independently, you will also have the CSA coming and binding. Interestingly, the CSA is not independently coming and binding, it comes as a complex which includes the uh, ubiquitin ligase 3 system that is made from COP9 and ROC1, and it also has a DNA damage binding 1 protein. So this will come and bind to the RNA polymerase and the RNA that has been formed. So this is how you can see is coming in. The XAB2 also comes and binds to stabilize this complex. In the meantime, because the RNA polymerase is stalled and CSB is able to bring in all the NER complexes, so you can see that you have the RPA coming and binding to the non-damaged strand, you have the TF2H coming and binding to the damaged strand with its with its XPB and the XPD um, coming and binding, and then you have the ERCC1 XRF coming and binding, and you have the XPG coming in. So there's an incision here, there's an incision here, this strand is removed, and then you have the DNA polymerase coming and synthesizing the DNA using this as the template. Once the repair is complete, because this strand is replaced, okay, by a new strand, so when the NER process is complete, then you can have the transcription starting again, and in the initiation of the transcription again, P300 and transcription factor 2s have a major role to play. So the only difference in the transcription coupled NER and the global genome NER is in the initial phases where CSA, CSB, XAB2 along with the HMGN1. This HMGN1 is responsible for making the DNA a chromatin again, or for basically associating with the chromatin, enabling the NER system to function properly. So let us come to the concluding remarks. NER is a wide spectrum DNA lesion repair system, as the XPC RAT23B identifies the damage by interacting with the destabilized duplex. And this destabilized duplex is in turn because of the presence of a lesion. So the GGR-NER initiates with the XPC RAT23B binding to the lesion, followed by formation of a Preincision complex. And this tends to bind to two endonucleases, which forms incisions on either side of the lesion. The TCNER is initiated with binding of CSA and CSB to the stalled RNA polymerase, making way for the assembly of NER and excision of the oligonucleotide with the lesion. The RFC, PCNA, DNA pol, and DNA ligase together as a complex help in filling and sealing the gap to complete the NER process. So NER with its broad spectrum substrate specificity is able to repair many DNA lesions, especially in repairing UV exposure DNA damage in both quiescent as well as replicating cells. Thank you.